Hey guys, guess what? You finally made it through Unit 2. This is our last chapter in Unit 2. And in this chapter, we're going to talk all about how we utilize information we've been talking about in the previous 5 chapters and work on experiments in the lab and test this information further to answer more questions about cell biology. This chapter is broken into 3 topics. And in this first topic, we're going to talk all about manipulating and analyzing DNA molecules. So we've already talked about a lot of these assays we're going to talk about here, but I want to talk about them in the context of the content we've been talking about so we can understand how we use these tools to understand the material that we've been talking about and how we move forward with the information. Here's our topic objectives, and as always, if you have any questions about them, let me know. Given the fact that you may or may not have used some of these assays, I fully expect that you may have more questions about them. So we're going to cover them briefly, but as I said, feel free to ask as many questions as you have about them because a lot of times you may not have had a chance to have hands-on experience with it yet. The most important thing when we talk about any kind of cell or lab assays is technology. Technology and science are intertwined so significantly that you can't separate them almost. A lot of the time we can't move forward with scientific discoveries until our technology catches up. And we're actually at this really great time right now where our technology is moving so quickly that we are actually in science not necessarily hampered too much by our technology. There are some areas that we are, but we have so much new technology to help us do these assays and understand this science that we actually have a lot of new techniques, and this is why we're discovering so much more about the cell lately. So it's really important to understand that. I put a lot of technique videos for you in Blackboard, so please let me know if you have any questions, but I encourage you to check them out. So let's talk about what we're going to focus into here for topic 1. There are 4 assays I want you to understand. The first is using restriction nucleases. The second is gel electrophoresis. And we're going to talk about hybridization, which we've already talked about before, but we're going to talk about it again. And then we're going to talk about blotting. So the first thing that we're going to talk about is restriction nucleases. Now, what are restriction nucleases? We've talked about them with regards to DNA polymerase when they fix a mistake in the replication process, or if something gets damaged, they come in and they'll pull out the broken nucleus or broken nucleotide. But what we do in the lab is we have these specific restriction nucleases that we've discovered or manufactured sometimes that all cut DNA in a very specific way. Each of them has their own cut. And you can see some examples of it here. They cut in 2 different ways. One's a blunt cut where they cut even on each side, and one's called a sticky end where they cut less on top, more on bottom, or vice versa. And it creates these overhangs that make them more sticky, and they actually re-anneal better together, or better when you use a sticky end. And that's why most of the time you use sticky ends, but sometimes you use blunt cuts. It just depends. Now, as I said, each of these cuts in their own unique way, and that's because it's looking for specific bases that it'll cut around, and you can see some of the examples here. So make sure you understand how this works and how these different examples work. Not necessarily, I will never ask you to know what EcoR1 does, but I want you to understand the differences between them. So what do we do with that then? So after we've cut some DNA, we will then run it on a gel electrophoresis. Now you guys should have seen this many times already in a variety of classes, and you've probably already done it in lab this semester as well. So what we do is we cut the DNA in our specific ranges, and so if we're comparing 2 samples, for instance, let's use our B201 example where we're looking for a suspect. We've taken samples of DNA from everybody, and we've cut them with a specific restriction nuclease. Every single one of them's got it. And so what'll happen is they'll get a unique set of bands. We run them on this gel, and as you know, DNA is negatively charged, so it runs towards the positive end. And it separates the pieces out by size. And then we can compare where the bands fall to see who matches, because we've used that same cut. We use the same technique when we're talking about foodborne outbreaks or other areas where we want to compare DNA quickly without actually sequencing it. This just allows us to see size-wise how different they are. Then we'll always stain it either using ethidium bromide or any other way to observe the bands. We can also then transfer this to a blot if we want to, and we'll talk about that later. So we've talked about hybridization before when we talked about the microarrays. Hybridization relies on the key concept that we can heat up DNA, break it into single strands, and then when we cool it back down, it'll come back together. What we do when we do this is we will heat up DNA, then we will expose it to complementary sequences, that are not necessarily its original complement strand, it's another one, and then let those reanneal together and come back together. And usually we've dyed the set that we want to come back together with. And so that helps us find those complementary sequences and identify whether or not that specific sequence of DNA is there. So how do we use this? So one example is in blotting. Now we've talked about Western blots where we use proteins. And in proteins we detect with antibodies, and that's because that's just how we detect our proteins. DNA is different, and RNA too. And so those blots are called the southern and the northern blots, where southern blots are the DNA blots, northern blots are the RNA blots. What we do is pretty much the same process as the Western blot. We run our gel, and then we'll transfer it to a nitrocellulose membrane, or sometimes there's other ones too, but nitrocellulose is kind of the mainstay. So now we have our bands on this new membrane. And then we'll probe it with DNA, and this will allow us to see specific sequences of DNA. So where the electrophoresis allows us to just see size of bands, blotting allows us to find specific sequences of DNA. So if we're looking for a specific gene, blotting allows us to do that. So this is just a quick overview of some of the ways that we manipulate and analyze DNA and RNA. So in this topic, please review those objectives. Let me know if you have any questions, and when you're ready, move on to topic 2. And then remember, we have a third one for this chapter, so we actually have 3 topics for chapter 10.